Hello everyone, and many thanks to the workshop organisers for the invitation to speak at this workshop on the surveillance and monitoring responses to highly pathogenic avian influenza. So over the past few months, we've seen a major change in the impact of avian flu on our wild bird populations. Normally, a range of different influenza viruses can circulate in wild birds, and these can be a source of major outbreaks of highly pathogenic flu in poultry. But in wild birds, these have tended to be linked to sublethal infections with relatively little impact on survival. But over the last 2 years, you can see that this has changed. Last winter, we saw large die-offs of wildfowl, for example, across the UK. And instead of reports of flu declining as we came out of winter, as we might normally expect, we've seen these ongoing incursions into a range of species, most notably the seabirds, but with very variable but locally catastrophic effects. And the seabird community has rallied as we might expect, asking what we can do. Where are the knowledge gaps? What data do we need? And how do we collect it? So we're now at a crucial stage where we want to move quickly from our immediate rapid response, which stopped many monitoring activities in order to protect vulnerable seabird populations, to one of more systematic and targeted surveillance with appropriate biosecurity to establish the information needed for future mitigation and conservation plans. And there's considerable scope to do this, building on what the community does already. So the workshop team have asked me to talk about some of the current approaches that are being taken to set the scene for where we go from here. Now, this is by no means an exhaustive overview, but are approaches that have come up in our conversations this season, so they seem like a good starting point for our discussions going forward. And just to acknowledge all of those have been part of these conversations. This includes colleagues at CEH who are our long-term collaborators on our avian disease work, colleagues at the University of Edinburgh who have been in the field collecting samples that they're now analysing. So a particular shout out to Fiona Greco and Hannah Ravenswater, and our health and safety team who have been advising on biosecurity, along with our Roslyn and AFA colleagues who are part of the recently announced FluMap consortium. And finally, NatureScot colleagues who have been responding to a continually evolving situation over this breeding season. So there have generally been 4 different types of approaches undertaken so far this season. And they are the sampling of dead birds, population monitoring, environmental sampling, and population sampling. So at the start of any outbreak, when the first signs of possible avian flu appears, the focus tends to be on the collection of sick or dead birds with designated labs to test whether they're positive for avian flu. This is an essential first step to confirm whether avian flu is present in a population, and it enables us to take steps to try and limit its impact. But viral samples can also potentially be obtained for sequencing to characterize infection and look at broader scale epidemiology. However, normally just a few birds are tested, and once avian flu has been confirmed, no further testing tends to occur for that species at that site, and there's no testing of surviving parts of the population. So this leaves open a number of questions. For example, how virulent is the virus for a particular host? Is the virus lethal, or is a larger part of the population infected but able to tolerate this infection? And what proportion of the population are infected at any given time point that could potentially transmit the virus to other individuals? And are there specific parts of the population more likely to be infected, such as juveniles that may prospect between colonies and who have yet to build up any acquired immunity? This type of testing can also bias our attention to those species which are most affected by the virus. We know there are other species often nesting in very close proximity to affected birds, so are presumably exposed to the virus, yet themselves appear to be relatively unaffected. So are these species reservoirs of infection, and what are the mechanisms underlying this variation in response. There may also be differences in detection probabilities of different species. A dead gannet on a cliff or in water can be observed from some distance, whereas sighting perhaps a dead shag on a beach in among seaweed or a puffin down a burrow is far less likely. Species also differ in their distribution, and that will affect how likely they are to be picked up for testing. So moving towards a more representative programme of surveillance across time and space would help us answer some of these key outstanding questions. And this is where the seabird community are already contributing. So the observational monitoring of populations already forms a large part of what we do. And this can start to tell us more about the wider impacts of flu at the population level and hence the broader implications of these outbreaks. It can also potentially identify routes of initial incursions into the population and where flu may be having most impact over different temporal and spatial scales. Movement data might also help to identify common sources of infection that birds might use away from colony areas and also onward transmission routes. And this might become particularly important in the coming months as we move out with the breeding season, at least in the northern hemisphere, and birds start to disperse or migrate away from their breeding colonies. However, the one limitation of purely observational sampling is that we can't untangle what effects are specifically attributable to flu and what might be caused by other pressures that seabirds are currently facing. So, for example, we're currently getting reports of young guillemots being washed up on the east coast of the UK. This also happened early last autumn. Yet of 142 birds that were tested during last year's die-off, none were positive for avian flu. So instead, changes in the usual seasonal distribution of fish are thought perhaps to be the cause, and therefore we can't assume novel die-offs are due to flu this year without further testing. Thirdly, there's environmental sampling being conducted. So this involves collecting fecal material or sediment samples where birds congregate for molecular analysis of both host and virus. And the advantage of this type of sampling is firstly that it's non-invasive, and it can be done outside times when the risk of transmission is high, and it can be used to obtain both viral and host genetic material. However, again, we need to think about how this data is collected going forwards. So, for example, simulation modelling has shown how pooled samples, when collected from below roosts of bats affected with Hendra virus, can lead to very different estimates of prevalence from those derived from individual-based sampling. So appropriate collection design and proofing with individual-based studies is key to interpreting future results. The virus can also persist in the environment for extended periods of time, so any sampling regime has to be undertaken with this in mind. There are biosecurity and legislative guidelines that need to be adhered to when handling avian flu viruses, particularly H5 and H7 viruses. Therefore, before considering any type of sample collection, discuss your plans with the relevant bodies both in your home country and field site location, and consider not just whether samples can be collected safely, but whether samples can be collected, transported and analysed to meet these requirements. So finally, perhaps the most useful type of sampling is the direct collection of diagnostic samples from across a population. This commonly includes blood sampling or swabbing for viral detection or antibody detection. And one of the advantages of this type of sampling is that it can potentially tell us about both current and previous infection, and therefore more about infection dynamics in both host and virus. Mechanisms underlying resistance and tolerance. And this type of sampling is particularly valuable when done in individually marked populations with long-term data, as it can then start to address some of the other missing parts of the jigsaw about which individuals may have been infected with different viruses and survived, how infection and immunity varies across the age structure of a population, and the impacts of infection on other key traits other than survival, that are important for our demographic understanding, such as breeding success or migratory movements. However, this is the most invasive type of sampling, so it comes with risks that have to be mitigated. So good biosecurity and field knowledge of the particular species that we're working with is essential. Again, this might involve samples containing a viral pathogen that can be both highly virulent to birds and has zoonotic potential. So again, there's a need to discuss any plans with relevant bodies for your particular circumstances. So these are some of the approaches that have been taken to date. Each, you can see, have their strengths and limitations, particularly when conducted in isolation. So where we can now potentially create real synergy is by underpinning our broad-scale monitoring approaches that we conduct across scale with targeted proof of principle intensive monitoring at key representative sites where we have individual-based long-term data. So perhaps as a starting framework for going forward, ideally what we want to do is firstly consider the questions we need to answer, because the type of sampling we do, where we do it, and when we do it will vary with the type of question being asked. Secondly, we want to consider future sampling regimes that combine these different approaches in the most optimal way to generate data that is representative, or at least enables us to account for sampling biases. Thirdly, we want to explore how we can standardize the collection of key data, and that's to ensure data is widely compatible for analysis across populations and aligns with broader reporting by national and international structures as avian flu operates at a global scale. And fourthly, we want to establish safe and biosecure ways of working that limits impacts on seabird populations, but enables the valuable information we gain from research and ringing activities to restart, as this is key to both understanding how best to limit any impacts of avian flu, but also the wider pressures that are still acting on our seabird populations going forward. So I hope that overview has been useful in terms of starting the discussion about how we co-develop the next steps from here. Thank you very much.